my pleasure to introduce our speaker for our PARC seminar today, who is Dr. Warren Beck, who's a professor at Michigan State University. Warren received his bachelor's in 1982 from Davidson College. Then he did a PhD at Yale with Gary Brudvig, well known to many of us here, studies of EPR and other topics on photosystem 2. He did Warren's really worked through top people in photosynthesis, because then he did a Miller postdoctoral fellowship period with Ken Sauer at Berkeley, followed by a position at Vanderbilt, and then he moved to Michigan State University in 1998, where he's now professor of chemistry. The Warren Beck group works on femtosecond nonlinear optical spectroscopy to study photophysical properties of photosynthetic systems, photosynthetic antenna, reaction centers, and model systems. Current focus, which I think is the topic of this talk today, is on carotenoids, energy transfer, and photoprotection. They also work on basic solvent dynamics and solvent relaxations of chromophore-containing proteins, including solvent reorganizations, and in response to electronic excitation. And some of this work involves coherent spectroscopy, I believe, which is a very important topic these days. And a lot of their work is nonlinear optical spectroscopy, where they use 2D photon echo and transient grating spectroscopy, which I haven't heard about for some time, which is I learned a lot about it at one point, but I think I've forgotten everything I knew. So it'll be good to hear about that if that's part of the presentation. So anyway, it's really a pleasure to have Warren here today and hear about his research. Warren.
Well, thanks, Dewey. I'd like to thank the PARC Center and Professor Lowe, who invited me. And I've enjoyed a short period of time walking across campus. Lovely campus, and the facility here is very impressive. And I'm very pleased to be here to tell you about some of the work that we're doing in my lab on carotenoids. So this is a brief summary of the kind of things that I'm thinking about. Sorry about that. Dewey mentioned some of these things, but I've been interested for a long time, since my days with Ken Sauer, in how photosynthetic light harvesting proteins work, in particular about how the architecture of the protein and the way that the chromophores are arranged spatially relative to each other and in the protein binding site, how that optimizes the system in terms of its quantum efficiency. And of course, people who work in this field, and probably there have been talks about it at PARC, about know that there's a current hot issue about the role of electronic coherence or delocalized excited states in systems like the Finna-Matthews-Olsen protein and other systems where there's clearly intermediate to strong electronic coupling. That's something I worked on a while back when in the phycobility proteins, alpha-phycocyanin and C-phycocyanin. And so I've been interested in that in a long time. Now the new project I'm going to tell you about today is in collaboration with Harry Frank's group at University of Connecticut, which has to do with radiationless decay and the nature of intramolecular charge transfer in the pyridinine chromophore in this system, the pyridinine chlorophyll A protein from dinoflagellates. Just in passing, the other project that my group has been working on for some time, Dewey also mentioned, has to do with dynamic solvation, how it is that, for say in this structure, cytochrome C, the surrounding protein and the coupled hydration shell water molecules respond to charge transfer and reorganization dynamics and limit or control redox properties. And I think this is of some importance also in understanding how proteins like light harvesting proteins can couple their conformational motion to electronic excitations. But the project we're working hardest on right now has to do with how the hydration shell, in this ND simulation, you can see the cloud of water molecules surrounding the protein, how those water molecules are coupled to the motions of the protein. That's a problem that's of current interest to the protein folding community as well. So that what I'm going to go to talk about today has to do predominantly with some new nonlinear optical experiments that my group and I have done on beta-carotene and paradenine to look at how their excited states can play a role in energy transfer. I would say as a newcomer to this area of beta-carotene and carotenoid photophysics that in reading all the literature, I sort of didn't have the impression of current literature how important the carotenoids are. I'm sorry, I pushed the wrong button. How the carotenoids play an important role as antenna chromophores for the mid-visible part of the spectrum. I had heard, of course, in my reading in the past about how carotenoids serve as in photoprotection and quenching excess triplet states from the bacterial chlorophylls and chlorophylls in the light harvesting proteins, and how they can play a role in scavenging of singlet oxygen. And of course, also lately, there's been a lot of discussion about non-photochemical quenching and regulation of the rate of energy transfer from chlorophyll to the reaction center, how carotenoids can play that. And in my association with the Brudvig group, there's a lot of interest back in the 80s about how carotenoids in photosystem 2 can play an important role in deactivating the photosystem 2 reaction center and charge transfer. So carotenoids play a very wide and essential role in photosynthesis. But what I underestimated is the sophistication and apparent strong role that they play in light harvesting. So here's in a nutshell, the problem about energy transfer from carotenoids to chlorophyll. They serve, as I mentioned, as a mid-visible light harvester. They strongly absorb in the 500 or 450 to 550 nanometer range, where the chlorophylls do not absorb strongly. So they can play an important role in absorbing the mid-visible photons, which are the strongest in flux at terrestrial level. And so they naturally have evolved to serve that purpose. But the problem, as is summarized in this diagram, is that they shouldn't be very good energy transfer donors to chlorophyll. Because of their symmetry properties, which appear to, even though they are distorted in the protein binding sites, they follow the selection rules for the C2H representation. And so that means that the strongest electronic transitions are from the ground state singlet to the second excited singlet state, which has this 1Bu plus label. So this is a strongly allowed transition because it goes from a Garada to ungarada state, and the parity goes from minus to plus. And that makes it a allowed transition for electric dipole allowed optical transition. In contrast, the first excited singlet state is a symmetry forbidden, at least formally, transition. And this is the state that is the lowest lying state that could serve as an energy transfer donor to the QY state of bacterial chlorophylls, the lowest singlet state. The reason this is an important channel is, of course, that the second excited singlet state of the carotenoids is very short-lived, probably less than half a picosecond in lifetime in most systems. So that means that even though it is lined up in energy usually to transfer energy to the QX state, the split upper next highest singlet state in the chlorophyll system, that energy transfer pathway is clamped by radiationless decay to the S1 state. The S1 state lives much longer, typically a picosecond or so, 10 picoseconds or so, so it lasts plenty of time to transfer energy to the QY state, but because of the forbidden character of its relaxation to the singlet state, this channel of energy transfer should not be very efficient. And so that's an interesting problem, yet we know that these carotenoids do serve as efficient energy donors to chlorophyll. So the problem I'm interested in, and my collaborator Harry Frank has been interested in for some time, among many things, is how the S1 state can serve as an energy transfer donor to chlorophyll using this QY channel. So there are many structures that you can look at. The structural biologists have done an impressive job over the last 20 years of crystallizing nearly every important example of light harvesting or reaction center structure in photosynthesis. So we now know where everything is. It's quite a change from when I was a graduate student and we didn't know where anything was. So it's nice to know. And you can look at the variety of light harvesting and even reaction center structures and find that carotenoids are typically found close to chlorophylls. And this is one way that energy transfer from a weak dipole strength S1 state can be improved. Work in the Fleming lab back in the late 90s showed that despite the possibility suggested by structures like this one from LH2, where the carotenoid seems to be almost in van der Waals contact with this pair of chlorophylls in the B850 part of the LH2 system, you would think that possibly Dexter or orbital overlap mediated energy transfer would be a possible mechanism. But experiments and calculations by Greg Scholes and the Fleming group suggested that Coulomb coupling, the ordinary downhill Fourier mechanism, dipole resonance energy transfer, can account for carotenoid to chlorophyll energy transfer in both the S2 and S1 channel because of the closeness of the carotenoid to the chlorophyll. If you integrate over the point-to-point couplings, there is still adequate, even though the transition dipole from S1 to S0 is weak for the carotenoid, it's being close-lived, close-spaced, the energy transfer rate can still be efficient. The yield of this system, if I remember correctly, is about 70% overall, or LH2, which is not as impressive as you would like, I guess. The PCP, or pyridine chlorophyll protein that I'm going to talk about in a second, does better, almost 95% efficiency. And there are possibly ways that the protein can optimize the coupling, especially in the S1 channel, by doing more than spacing the chromophores close together. And the hypothesis that emerges from my reading of a lot of the current literature is that binding site control of the ground state conformation and possibly, which I'm going to raise this today, excited state conformational motion can enhance the S1 state transition dipole by mixing the carotenoid S2 and S1 states. So in other words, the S1 state can gain dipole strength by borrowing it from the S2 state, principally by bending or twisting the conformation of the carotenoid. This is part of the work that comes out of Bob Burge's laboratory at UConn, which is part of the collaboration with Harry Frank. So here's the system I mentioned, pyridine chlorophyll A protein. This one is from amphidium. And the thing that is unusual about PCP protein is that instead of using chlorophyll as the main light harvesting chromophore, this system uses a modified carotenoid called pyridine as the main light harvesting chromophore. Its function is to transfer energy to chlorophyll A. You can see in this exploded view of one of the subunits, carotenoid and chlorophyll complement, that there are 8 of these pyridine carotenoids packed around 2 chlorophyll A's. And the calculations by Damjanovic in Carl Schulten's group about more than 10 years ago now suggest that 4 carotenoids function to transfer energy to the chlorophyll A's, and of course chlorophyll A could do chlorophyll A to chlorophyll A energy transfer and eventually to photosystem 2 or photosystem 1 by spillover, I suppose. I'm not sure if it is actually known the actual pathways that pyridine and chlorophyll protein feeds, but naturally the chlorophyll A can act as a good donor for chlorophyll A and other light harvesting and reaction center proteins. Now the picture that comes out of these calculations from the Schulten lab is that because of the strong transition dipole strength for the S2 state, the 4 pyridines are probably in the intermediate or even strong coupling regime. So there's a possibility of energy transfer coherence in each domain. And this might be a possible mechanism to augment or improve the probability of energy transfer from the S2 state. But the thing that has interested people for some time is the fact that paradenine has apparently strong charge transfer character in the S1 state that gives it a strong solvent dependency and a strong lifetime dependency based on the solvent. And the origin of this, from a lot of work in several laboratories, but some in Harry Frank's laboratory, is this lactone ring, the 5-membered oxygen heterocyclic ring, and the carbonyl group, which is in conjugation with the main polyene conjugation. And there have been a lot of studies in Harry's lab about what happens when you move the position of the lactone ring up and down the paradenine polyene, and also in other carotenoids that have carbonyls. The key is the carbonyl. The carbonyl affords the carotenoid an unusual sensitivity to the surrounding solvent, and there's the strong supposition that there's an intramolecular transfer of charge down the polyene that creates a large dipole strength, a larger than normal S1 state probability for emission and energy transfer. Now, this is a point of current controversy. How is the charge transfer character in paradenine produced? There are at least 3 mechanisms that are being discussed. 2 of them, the left and the center one, involve actual transfer of charge. So the S2 state delivers by non-radiative decay on a sub-picosecond or approximately picosecond timescale population to a local the non-ionic character or non-charge transfer character region of a configurational potential energy surface that's coupled to charge transfer. So that in one limit, the donor and acceptor regions of the potential surface are strongly coupled. So this is said to be an adiabatic mechanism. The crossing from local to charge transfer character would be a high probability. Whereas in this non-adiabatic or diabatic picture, whether you prefer double negatives, it's your choice, the coupling is less strong between the local non-ionic and charge transfer regions, and the probability of jumping from this region of the surface to that one would be lower. So you might predict a different kind of time evolution for these 2 different models. This model would suggest that once formed, the S1 state would transfer very quickly and with high quantum yield the charge transfer region of the surface that is labeled ICT here, or intramolecular charge transfer. Whereas here you might expect an equilibration of population between the 2 states and a lower probability of transfer, a slower, maybe temperature-dependent rate, temperature-dependent yield. Now the alternative, which is the one favored by Burge and Frank at present, at least that's my understanding, is that the S1 state and the charge transfer state are, in their language, one and the same, meaning that this structure has an intrinsic charge transfer probability or property, and that the S2 state delivers population to it, perhaps, as I'm going to suggest in this hypothesis, that it's excited state dynamics, structural dynamics, that prepare this charge transfer character that is realized once non-radiative decay from the S2 state occurs. So that's what I'm wondering about, is if we could study using nonlinear spectroscopy to see how the S2 state evolves, and then eventually, probably using 2-dimensional spectroscopy, follow the evolution of the S1 state to see if we could work out how this charge transfer character forms. So the methods we've decided to work with are the relatively current 3rd-order methods that include the stimulated photon echo and transient grating spectroscopies that are the basis for the modern 2-dimensional spectroscopies that people do to study light harvesting. So I'd like to tell you briefly about how this is done, because a lot of people, myself included, since this is recent for my group, the development of the technology to do this. Quite interesting area that I think marks a significant growth in the capability of nonlinear optical and femtosecond spectroscopy over the last 15 years. A lot of the work I'm going to talk about is the product of David Jonas's work at Colorado Boulder, who I think really seminal, extraordinarily impressive work developing the technology called spectral interferometry and the application of 2-dimensional methods derived from NMR correlation spectroscopy methods to come up with these new techniques that are now becoming routine enough that my laboratory is starting to try to use it. So all these methods are based on 3-pulse experiments that are really strongly derivative of those in NMR, the Hahn echo sequence. And so the idea is that 2 pulses are used to first prepare and then destroy an optical coherence, an oscillating charge density. So one interaction with the optical field, this pulse K1, creates a coherent superposition state, which is then allowed to evolve for a short period of time, typically a few hundred femtoseconds or less. And then that coherence is destroyed to make a population. The population then evolves for this longer time period, big T in most people's experiments, at least the Fleming group uses this terminology. This is a population time. And then the 3rd pulse arrives and it recreates a coherence and reconstitutes, so to speak, the stored coherence that was destroyed at the 2nd pulse. Then at a later time, a signal is emitted, depending on whether or not there's any phase coherence remaining after this coherence time, tau, between the first 2 pulses. The signal can be an echo or a free induction decay, just a prompt decay of the coherence that is produced by the 3rd pulse. Now, in the experiments I'm going to talk about today, what are called transient grating experiments, this time tau is set to 0, so the first 2 pulses arrive at the same time, but the momentum vectors are different. This allows us to select, using the 3rd pulse, a unique direction for the emission of the signal pulse. The advantage of that is that allows us to cleanly use a weaker pulse, which typically arrives first, called the local oscillator pulse, which interferes with the signal so that we can read out the nonlinear optical part of the signal, the 3rd order response, using a Fourier transform method. So just briefly, the apparatus we use is based on a design mostly from the Fleming group. There's a paper by Tobias Brixner in 2004 in the Fleming group, which described the use of wedge-shaped prisms and a diffractive optic, which I'll explain briefly, to prepare and control the time orderings for the 3 pulses. In our setup, and their similar design was developed almost at the same time by R.J. Duane Miller's group at Toronto. Now the machine we built is strongly derived on one built by Andy Moran in Norbert Scherer's lab at the University of Chicago. And Andy has been very helpful to us by explaining how this thing works and sharing parts of his software. So it's been extremely helpful in this project. Our setup uses a high rep rate Ti-Sapphire laser. It's a coherent Mira Reg A system. So we have 250 kilohertz, 50 femtosecond pulses from the Ti-Sapphire that feeds a conventional collinear OPA that makes 35 to 40 femtosecond pulses. We've tuned it for use with the carotenoids and similar chromophores down to the extreme blue-green region of the operation, so it's about 520 nanometers typically. So now the details on this system are interesting to me. The first and 2nd pulses in the pulse sequence and the 3rd and local oscillator pulses are separated from the OPA pulse using a conventional interferometer and a beam splitter. And so they have, the population time is already set by conventional means, but the coherence time tau is set using techniques that Brixner developed in the Fleming lab. So the way this works is both the 1st and 2nd pump pulses and these probe pulses are split themselves into 2 pulses by a diffractive optic, a transmission diffraction grating. And then using this scheme that Moran and Scherer put together, which is also used in the Scholes laboratory in Toronto, I understand, the 4 pulses that emerge from the diffractive optic are then imaged onto a spherical mirror so that they are aligned in the corners of a square. So this older diagram uses a slightly different picture, but these 3 pulses are the ones propagating between the 2 curved mirrors in this design. And so this mirror here re-images the focus from the diffractive optic onto the sample in an analogy to here, where the 3 pulses get focused and then are inverted when they come out of the sample. And they are then recollimated by the second curved mirror in lieu of this lens in this picture. Now the key point is that due to momentum conservation from the first 3 pulses in the transient grating or stimulated photon echo sequence, the signal emerges on a unique unique vector. So depending on whether pulse 1 or pulse 2 are in front, it emerges on these 2 slightly different color beams that are at the corners of the cube, the square picture of the propagation of those pulses. So we can arrange to collect that pulse and image it into a monochromator or a spectrograph and to hit a CCD. To read it out, to read out the signal in order to get all the information, the heterodyne or optical heterodyne detection scheme is used. So we then align this 4th pulse, which came from the other pulse split off by the diffractive optic, to line up so that it goes directly along the K signal direction. So that way we can interfere them in the spectrograph and read out the nonlinear optical signal. So this is a depiction of that process, which is part of the work, I think, that David Jonas worked out in his laboratory around the year 2000 or so. It's called Fourier transform spectral interferometry. So the idea is that in the absence of that local oscillator pulse, what you see at the detector emerging along the signal direction in this experiment is a unmodulated spectrum, which looks sort of like the laser spectrum, but it's been changed by, of course, the interaction with the molecule in the sample. And when the probe or local oscillator beam, rather, is allowed to overlap with it spatially, you get an interference pattern, which you can then use a inverse Fourier transformation from frequency domain to time domain. What you see is that there's a broad spectrum pulse at 0 time, which corresponds just to the laser shape. But then the interference fringes that you see correspond to a pulse at a later time, and this time corresponds to the time difference between the local oscillator pulse, which, as I mentioned, arrives first in most of these experiments, in our setup about 800 femtoseconds before the signal pulse, the 3rd pulse in the system. So we can window that and then back transform it into the frequency domain. And now the signal is complex or oscillatory. We apply a phasing process where we multiply this complex signal using the time of the local oscillator and an adjustable phase, which we have to determine using either a probe signal or a signal in a dilute, a neat solvent, so that we can determine the absolute phase factor. And that allows us to isolate 2 components in this signal: the real component, which corresponds to the absorption signal, or photobleaching stimulated emission, and the imaginary component, where this signal is pointed down due to our choice of phase factor. The imaginary component is called the dispersion. To give you an idea of what these 2 signals are, I show you this diagram, which is based on a classical picture for the work done by an oscillating electric field, say a ray of light, on a set of dipoles. And this picture is sufficient to understand what the absorption and dispersion components of the signal are. So imagine that there is a resonant frequency for the system that you're in your sample. Here it's at this frequency here. And so the component that's in phase with the oscillation of the electric field is called the dispersion. And you can see that at low frequency, say it's 0 frequency, as we scan the frequency of the light source, the dipoles in the sample are able to keep up with the oscillation of the electric field until we get to the resonant frequency of the system, at which point a phase shift occurs. A dramatic phase shift occurs. This occurs as the electric field starts doing work. The oscillators in the sample start absorbing some of the energy of the oscillating electric field. And that causes this change in sign of this dispersion component, which is in phase with the oscillating electric field. The absorption character, or the work done, is pi over 2, or in quadrature with the electric field. And so as most people report, the nonlinear optical signal that you get is oscillatory, and therefore it's complex. You can describe it using two different terms, either a in phase or out of phase term. Those are two components. Or you could have the amplitude and its phase. And for the carotenoids, as you'll see, the choice is to use the two phases, components, the absorption and the dispersion. Okay, so let me give you a quick run through of what some of these signals look like. So rhodamine 6G is a good molecule to use if you've tuned your OPA up to work with carotenoids, so you can test out your system. And it's a fairly simple system in that it only has one operating electric excited state, so we can pump it and then observe ground state depletion, stimulated emission, and excited state absorption to some higher singlet state. This is what you can do in an ordinary pump probe experiment. There's no radiationless decay to worry about from the S1 state, and so the character of the signal that you get is easy to understand. So here is a picture of the real component of the wavelength resolved transient grating signal that we get from spectral interferometry. And so it shows at 0 time the depletion of population from the ground state and the production of stimulated emission and some excited state absorption character. And with time, you can see there's a shift of the spectrum at a really short time out to the first 50 femtoseconds or so that involves dynamic solvation. This is in methanol solvent. If we slice that spectrum at different delay times, you can see the loss of the ESA character and the shifting of this stimulated emission portion of the signal. The signal includes photobleaching and overlapping stimulated emission components. So that describes a dynamic Stokes shift, which you can read out directly from the integrated transient grating profile, which I show here. So here are I'm using color coding, although it's hard to see here. There's red for the absorption part, and our choice of phase for the dispersion made the signal go down. And the modulus is the amplitude of the field. This would be the square of these two components added together, the square root of the square. And it would be what you would get in a conventional or homodyne transient grating experiment, because the detector is usually a square law detector. So you are measuring the square of the electric field. What the advantage of the heterodyne technique is that you get the two components separately. Here, and this is the point of showing you this, there's no interesting information in having the absorption and dispersion separately. It looks just like the homodyne signal would be. And the decay here is mostly attributed to dynamic solvation. There's a component at about 40 femtosecond time scale followed by a couple of hundred femtoseconds decay. And then you have long-lived population in the S1 state. So this is very interesting. I wanted to show this to you just so that you'll see how different the dispersion and absorption signals look from carotenoids. I should point out, I forgot I made a point here to say that the absorption and dispersion components have the same shape or temporal profile when all of the pathways that deactivate the excited state and give rise to the nonlinear signal originate from the same resonant excited state. And this is something that was established by Schutt and Fleming when they first worked out the heterodyne transient grating techniques in the Fleming lab in 2001 and 2002 or so. So there's nothing new here. Now in beta carotene, quite a bit more interesting possibilities. So in our experiment, we tuned, let me get back here, I'm sorry. We tuned the OPA to coincide with the lowest part of the absorption spectrum, so we're preparing the S2 excited state essentially vibrationlessly. And so what you might expect in the orthodox scheme for the 3 electronic states that would be involved is that we would prepare the S2 state, it would do a non-radiative decay relatively quickly to the S1 state, and then a non-radiative decay to the ground state. The timescales would be on the order of 500 femtoseconds or less, and 10 picoseconds. These are typical times for carotenoids. The result is quite a bit more complicated than in the rhodamine case, because you'd already expect it to be more complicated, because the S1 state has strong, even though it has weak transitions to the ground state, it has strong transitions to S2 and higher excited states because they're already known from probe experiments. So we expected to see quite different absorption and dispersion profiles, but maybe only 2 different relaxation times in the modeling. And we got quite a bit more complicated picture. So here is the short time region of the integrated transient wavelength integrated transient grating profile. The absorption signal does a prompt rise when the laser pulse arrives, the pump pulses arrive, followed by a fast decay that has 2 components on this time scale, about 15 femtoseconds and 150 femtoseconds, that lead to a long-lived, relatively long-lived S1 state derived excited state absorption signal. So here we have the transient grating signal goes to a negative going signal because there's a decrease of light as the Feynman pathways involving excited state absorption are driven as the S1 state is produced. Now the dispersion signal looks quite different, and in our choice here of benzonitrile, there's a large solvent-derived component that we worked out by doing solute concentration dependencies, so we could work out what part of this dispersion signal arises from the solvent. The one advantage of the heterodyne technique is that it is usually possible to separate the contribution of the solvent from the contribution of the solute, especially in that the solute is non-resonant. The absorption channel is essentially pure, and so we can model it explicitly. Notice that the dynamics of the S2 state are largely hidden in the modulus signal. The ordinary homodyne signal would not contain very much information about the initial relaxation. So the advantage here is that we're going to get a good look at the initial relaxation of the S2 state. Now on the long time scale, the interesting thing that we observe is that the absorption ESA signal and the decay of the dispersion signal occur with different time constants. The absorption recovers in 12 picosecond time scale, which would be on the order of the known S1 state lifetime. But the dispersion occurs more rapidly, about 9 picosecond time constant. So it is apparent that the absorption and dispersion are looking at different electronic states or different populations at least. Now, the picture that comes about here that we propose, and this is based on some work that was published about 10 years ago. I have a citation. Richard Cogdell's laboratory in particular published a very important science paper in 2002 using very short pulses. They worked on beta-carotene and observed intermediate states that were produced promptly after the formation of the S2 state. And our results are essentially in agreement with theirs, even though we're using different techniques. The S2 state appears to decay to an intermediate state in about 15 femtoseconds, and then this state decays to the S1 state in about 150 femtoseconds. So this state, this intermediate state is probably the state that gives rise to fluorescence. And also it's the one that serves as the donor in the energy transfer to the QX state. Now the S1 state recovers in 2 steps using in our picture. Initially a dark state is produced in about 9 picoseconds. This is what the dispersion channel senses, and I'll say more about this in a moment. And then the final ground state recovery is sensed in the absorption channel, and that takes 12 picoseconds. So there are a number of different possibilities to accommodate these 2 timescales. But one possibility is that we're producing some kind of ground state conformational population, which is distinct from the ground state, or it could be a hot, vibrationally excited ground state. Both possibilities are still worth considering. And you can see, I won't go through all this. I'm a little bit running late here. I always talk too much. The slices of the transient grating signal give strong evidence for the SX, this intermediate state. I've already given the assignment. It's called the SX state. You can see the decay of excited state absorption character from the initially S2 state character spectrum that we see out for maybe only about 20 femtoseconds, and then start evolving a strong ESA character to the red of the main photobleaching region. You know, it should be noted that this is a strongly whole-burned character spectrum because we're only exciting the lowest part of the carotenoid absorption. So these spectra are narrower than what you would see with a shorter pulse. And on a longer time scale, this spectrum here, this intermediate character spectrum that doesn't have much ESA in the blue, but you start developing a blue shifted ESA character in here in the red, to the red of the spectrum. This is what is characteristic of the S1 state. And you should keep in mind that it's known that the S1 ESA spectrum, excited state absorption spectrum, extends from this region at least to the farther to the red, and we're limited by our spectral laser spectral bandwidth to just see this portion of it. So here's the picture that I favor, and this is based on thinking about the timescales and some of the work here, this paper in Science that I mentioned, Richard Cogdo and Hashimoto, who has worked subsequently on vibrational coherence from the ground state and beta carotene. The timescales are compatible with this work, but I think the picture that comes about is somewhat different, still speculative, I want to make clear. Now, I suggest that the front Condon structure prepared directly by excitation of the ground state predominantly prepares this SX state on the order of 20 femtosecond timescale or less. This is about the same timescale that these fellows here in the Science paper derive from shorter pulses. The nature of this is likely to be an excited state displacement, just a vibronic displacement. And the character of this has to be stretching mediated from the front Condon region because the resonance Raman spectrum is dominated by carbon-carbon and carbon double bond carbon stretching modes. So what takes the system predominantly away from the front Condon structure, which is vertical from the ground state, are stretching motions. But I think twisting or bending would be but twisting would be the most efficient way to explain why this state here, the intermediate state, is dark from the ground state. And even so, its character is still close to what it was in the S2 state. So the symmetry label, that's all up in the air. But whatever it is, I strongly suggest that this is a structural displacement that occurs simply by adiabatic passage from the front Condon structure to a minimum on the ground state over a low barrier. And this barrier height could be both conformational but also derived from the surrounding solvent. Now, the radiationless decay then occurs in two steps. The first step would be this 150 femtosecond time constant step that would prepare the S1 state vertically from this SX or conformationally displaced state. SX state is the label that was proposed and has been used by these fellows and has been used in the carotenoid literature. So I'm calling it that, even though it may well be that what some people call SX may not be what I'm observing in these experiments. That said, the structure produced in the S1 state is derived from whatever happened in the S2 state. So it's not the structure in the front Condon region, it's something else. And then the relaxation to the ground state would involve another vertical or radiationless process that would prepare a not the front Condon structure, but a different structure initially. Now, I think this conclusion is consistent with this paper, which it's not a different paper here now, from Harold Kaufman's laboratory, and Lux and Christensen were involved in doing some calculations. On beta carotene, and I think they suggest the S star state, which has been widely discussed, is this conformationally relaxed state that's yielded in this picture from whatever SX was vertically. So this state would take some time to recover back to the original ground state by either crossing over a barrier or by vibrational relaxation. And the time scale is compatible with either, I think. 12 picoseconds is not a bad time to use as a rule of thumb for vibrational relaxation. So that's why this is a reasonable picture. Now, Heterodyne seems to follow similar excited state dynamics in the experiments we've done so far. The character of the absorption and dispersion profiles are quite similar to those of beta carotene, although the modeling we have suggests that this SX intermediate that is produced on a slightly slower time scale, 30 femtoseconds rather than 15 or so in beta carotene. This state has an net excited state absorption character over our laser spectrum, whereas beta carotene was still net photobleaching stimulated emission. So the character of this state is, probably shifted. This SX state is somewhat shifted spectrally compared to what it is for beta carotene. This is in methanol, and we don't have any complications from methanol in the dispersion signal in our experiments. So we can model the dispersion directly. We used a global profile to model the absorption and dispersion and to get these two components. And you'll note that the SX state that was produced lasts quite a bit longer in this picture, 615 femtoseconds, which is on the order of the time constant that people attributed to the S2 state in Heterodyne, I think, previously. So what we're suggesting is that the excited state of Heterodyne evolves rapidly to make the state that is then taking care of energy transfer to the S2 state. And similarly, on a longer time scale, the absorption and dispersion profiles decay with different time constants, similarly spaced, slightly shorter time scale than beta carotene in benzonitrile. This is in methanol or Heterodyne. So it seems to be a similar idea, similar picture for the radiationless decay and the photophysics. Now, if we compare, this is all I'm going to say about PCP because we're still getting these experiments going on Heterodyne and chlorophyll protein. We've done some transient grating experiments, and we're beginning to model the energy transfer pathways that will quench the Heterodyne and excited state, both in this XS, SX, and S1 state. And so we've scaled them here, the same optical densities, same laser pulse energies, so we can directly prepare or observe by inspection the implication of the contribution, rather, of energy transfer in both the S2 state channel and in the S1 state channel. So you can see first that, there are some notable differences. The first difference is that the SX state seems to form significantly faster in PCP than in Heterodyne. So it's possible that the protein's conformational control of the chromophore allows it to prepare this state faster than it does in solution, where a Maxwell-Boltzmann distribution, probably mostly planar conformations, are featured. The other thing you can see is that the populations are quenched. The dispersion signal, which is mostly character of S1, decays much more rapidly in PCP than it does in Heterodyne. This is the modulus is even more clear. But it's important to recognize that the initial state, in PCP, this is a lot less population early on than it is evident in Heterodyne. So it's clear that the SX state is being quenched as well. And the work that Harry Frank's lab and others have done suggests that about 1/3 the quantum yield of energy transfer goes through the S2 channel. And so it's this long-lived state that Heterodyne produces, 500 femtoseconds or longer, is a good energy transfer donor. So that sort of covers the picture that we have at present. The suggestion I have is then that this ICT character that forms in Heterodyne and that affords the S1 state its unusual lifetime and solvent sensitivity arises from this excited state dynamics, which probably is some kind of stretching or then subsequently twisting change in conformation of the chromophore. And that this state relaxes vertically first to the S1 state and then to the ground state and gives rise to the dynamics that people have observed previously. So the intramolecular charge transfer character is, in this picture, controlled by changes in conformation that arise very rapidly on the excited state surface. Now, two-dimensional spectroscopy with broader band pulses should permit us to get even more information. So what I've shown you here is some early single scan experiments on paradenine and methanol, which are designed just with the limited bandwidth we have now to follow the changes in correlation function that arise as the system evolves rapidly from the S2 state to this putative SX state. And so we can see that at early time, this is a very diagonal looking spectrum that evolves very rapidly around, looks certainly more spherical, as if a broader range of conformations is explored at this early time. But as time goes further, we see it assume another diagonal state. So this 200 femtosecond delay is highly correlated with the initial state. This is an interesting result because it suggests that we should be able to use broader band two-dimensional spectroscopy to watch how the initial S2 state is correlated with the time evolving S1 state as measured using cross peaks in the excited state absorption part. So that will require us to have probably 10 femtosecond pulses that span the 500 up to maybe 620 nanometer range. And so we've started constructing a non-collinear OPA, NOPA, to do those experiments, and those are sort of in progress. So I'd like to acknowledge people who have helped us in this project. Of course, Harry Frank and his super technician, Amy LaFountain, have been very, very supportive. Amy made the samples for us in this, all the work that I've talked about today. Andy Moran at UNC Chapel Hill, very helpful in helping us figure out how to build the two-dimensional and transient grating spectrometer, and shared with us a lot of his MATLAB code and was very patient as we kept asking stupid questions about how to do this stuff. And my colleague Gary Blanchard at Michigan State University loaned us his CCD camera, which is what we used in these experiments. The students who did the work, led by Michael Bishop and Soman Ghosh. Nolan Shepard is in Norbert Scherer's lab now at University of Chicago as an undergraduate in the lab. And Jenny Jo Mueller is a third-year student supported by a United States Navy duty under instruction posting. So she's about to finish her PhD on an accelerated program. And the lab was supported through a lot of this time period on a grant from NSF Molecular Biophysics. Thanks very much. So the question was about the heterodyne spectroscopy. How do you know that the local oscillator, which arrives first in the pulse sequence, doesn't have any impact on the subsequent photophysics that you detect? Is that reasonable paraphrasing? Well, the local oscillator pulse is extremely weak. It's attenuated 4 orders of magnitude using neutral density filters compared to the other 3 pulses. So its contribution to the actinic photochemistry is very small. But the other thing that's important is that the way we at least align this experiment is we block the local oscillator pulse and then optimize the overlap spatially of the 3 pulses in the experiment to generate a homodyne transient grating or echo signal. And once you get that lined up into the detector, you unblock the local oscillator. And it's not even aligned perfectly compared to those other pulses. All that matters is that it overlaps through our spatial filters and goes into the spectrograph so that it then makes an interference pattern. It's probably not actually overlapping spatially with the other 3 pulses in the sample. And so it's a weak pulse and it's crummy in its alignment. And yet, if you are restricted to the spatial region where the homodyne signal is, you can detect the heterodyne interference signal. And that's the part that we obtain and isolate using Fourier transformation. So we know that we're getting pure heterodyne signal in this algorithm that Jonas and others developed. So it's a very clean experiment, I think. That's a good question. The question was, can you do an experiment, say, at cryogenic temperature to slow down or control the solvent medium's contribution to reorganization around the chromophore? That's a good question. We haven't done any experiments like that. The barriers are in the excited state are unknown. It very well may be not really a barrier. To have something so fast take the front Conan structure and make a structure that is distinct spectroscopically, where it suggests that if there is a barrier, it isn't a large one, at least at ambient temperature. And of course, the position of that SX or intermediate minimum compared to the front Conan minimum is unknown. And the nature of the coupling between that state and the SX state is unknown as well. But the idea of using solvent viscosity or temperature to control the dynamics is a good one, and that's the kind of thing that people should do. We should try to do it. It is feasible to do this kind of experiment in a cryostat. The pulses are really weak. They're, in our experiment, they're 1 nanojoule total. And Greg Engel in particular has developed some really nice techniques using single pulse methods to do the whole two-dimensional experiment. So the amount of photo damage that you would do on a cryostat sample would be small. It's feasible to do these experiments in a static cryostat, that's for sure.
Going in the opposite direction, you see a larger contribution of the vibrational excited state would be rather than pumping 0, 0, your estimation of higher manifold or other operations come in another electronics.
Yeah, the question that Dewey asked was, what would the outcome be if you tuned the laser spectrum so that you selected a different vibrational energy level in the S2 excited state? Or perhaps the other thing you would do if you tuned is you would select a different group of ground state conformers from beta carotene. The paper I cited there studies the spectrum of these conformers, and there are a few kilocalories per mole in barriers in the ground state. Of course, in the excited state, these barriers would be less, a little bit less, you'd think. Yeah, but we haven't tried any of those experiments. I actually think it would be very complicated, though, if you started tuning the spectrum of conformers by tuning. You'd like to have pure selection of vibrational energy level, but you're also going to get different internal conformers. And so I bet the dynamics are different in the excited state just from those molecules. So it would be very entertaining to do. I think what you'd have to do is couple those studies with some computational studies of the distribution or spectrum, as I called it, of conformers, so you would know what populations you were producing in the excited state, so that you could try to decouple the vibrational level preparation from what the structure of the chromophore was. Of course, in the proteins, the conformers are apparently much narrower because they're selected by the binding site. And so it's like in pyridinine, the suggestion, rather in PCP, the suggestion I made, which is probably premature, but we'll see, is that the protein has selected a conformer in the ground state which is distinct from the one preferred in free solution. And the dynamics were different. So that could be either due to the amount of vibrational displacement in the excited state that would have to be made to make the SX state, or it could be that you, due to the front Conan factors, you'd get a different vibrational level prepared optimally. I don't know the answer to either of those possibilities at this point.
So my question, to answer the question, I was thinking also in terms of just adding excess electronic energy so you would produce more of the vibrational state. In principle, vibrational relaxation is the distribution of vibrational levels.
Right.
The red ones are going to go away faster than the blue ones. I don't know if you can see it. You're already distinguishing 9 and 12. If you actually look at that as individual wavelengths and walk across, you see a progression like that that's indicative of the vibrational distribution, which is ultimately probably
So I'll summarize that as a long question, but the suggestion, let's see if I can get it right. The suggestion was that you certainly should expect to see a different non-Boltzmann population of vibrational or vibronic levels in the excited state that you prepare vertically. And you ought to see a difference in the rate of relaxation to this intermediate state dependent on what the distribution is. And that experiment, you really should see that if my picture is correct. And yeah, I don't know. That's something we'd like to do. One of the technical challenges is, of course, that we're at the very end of the tuning range of the OPA, although the NOPA systems, if we can make a pulse that's 20 or 15 femtoseconds, a fairly routine bandwidth from a NOPA